The entirety of medical biochemistry operates as one highly predictable, interconnected physiological network, driving every cellular function. Every metabolic process maps back to 7 core branches, spanning from microscopic molecular genetics up to systemic clinical diagnosis. These sectors provide the logical framework needed to trace a single cellular event through the entire system to a specific patient outcome. The system's rules are set at the microscopic level. Genetic information flows in a fixed sequence: DNA replication, transcription into RNA, and translation into functional proteins. This graph illustrates Michaelis-Menten kinetics, where reaction speeds are governed by maximum velocity, Vmax, and substrate affinity, Km. Inhibitors alter these parameters predictably. Competitive inhibitors increase the apparent Km by blocking the active site. Non-competitive inhibitors lower Vmax by reducing the number of functional enzymes. Cellular dysfunction and disease states follow when these fundamental kinetic rules are disrupted or inhibited. This map shows the coordinated push-pull system of hepatic carbohydrate metabolism. In the fasted state, the liver reverses energy flow, bypassing the irreversible blue steps of glycolysis to generate new glucose through the green pathways of gluconeogenesis. This chart illustrates the Cori cycle, which maintains the energy loop during anaerobic stress. It traces lactate produced by exercising muscle traveling to the liver, where it is recycled back into usable glucose. Lipid metabolism follows a similar reciprocal logic. On the left, dense fats are broken down through beta oxidation in the mitochondria, while the right column shows their reduction and synthesis in the cytoplasm. This pathway maps how those lipids move through the bloodstream. The liver packages triglycerides into the DLL for peripheral delivery, while HDL facilitates reverse transport, returning excess cholesterol to the liver for excretion. The balance between these carbohydrate and lipid pathways dictates a patient's overall energy homeostasis and their long-term cardiovascular risk. Metabolizing these energy sources produces nitrogen waste as a byproduct. The urea cycle constantly detoxifies this ammonia to prevent systemic damage. When these biochemical pathways fail, damaged cells leak specific enzymes into circulation. Necrotic heart muscle releases troponin I, while hepatic injury is marked by elevated AST and ALT levels. Identifying these blood biomarkers allows for the precise diagnosis of which underlying metabolic branch has collapsed. During physiological stress, these modules operate as a single circuit. Energy generation, waste detoxification, and biomarker release happen simultaneously across the entire network. Connecting these individual pathways transforms biochemistry from a list of abstract reactions into a concrete roadmap for clinical diagnosis and treatment.